Thanks for joining us for the second part of this morning's session. So we heard a lot about some of the background theory and mathematics underlying metabolic modeling from our first two speakers this morning. And now we're going to move towards a little bit more of the applied, how to use the applications that implement these tools in KBase. And we're going to be hearing from Janaka Edirisinghe, am I saying that even close to right, hopefully, and Chris Henry. Both Janaka and Chris are from the Argonne National Laboratory, which is near Chicago. And they work in the bioinformatics group there and are very involved in the KBase development. So the first speaker is Janaka, and I'm going to let him take it away.
Thank you, Tim, for the kind introduction. And everybody can hear me, I guess. And so I'm a computational biologist at Argonne. And today, I'm going to talk about the microbiome modeling pipeline in KBase and how it's address important scientific questions using these tools. So before I begin my presentation, I want to acknowledge today's TAs that help students during the hands-on session. And so please utilize these workflows and ask questions and learn at the end of the day so you can help your projects. So to properly orient everyone here from day one, what we have done is we had samples and we had sequencing reads from the samples, and we have used apps like FastQC to assess the quality, and then we trim reads to remove the adapter sequences, and then we use tools like CAIU, which works on reads for profiling taxonomic classification, and then we used assemblers like MegaHit to assemble these reads into config, which we call the metagenome assembly, and then we used binner like MetaBed2 or MaxBin2 so that we can bin these configs into individual bins. And then we use the quality assessment tools like CheckM to assess the quality of the bins. And once we select the high quality bins, then we can infer genome annotations. We can make inferences to those sequences, identifying genes, the functions that's where we use tools like DRAM and the GTDB. So for today's workflows, we'll start from the metagenome assembly and also bins. So today's workflow consists of that we use the metagenome assembly, and then we annotate this metagenome assembly using Rust annotation pipeline in KBase, and similarly we also annotate these bins that identified from this sample, and then annotate with the Rust annotation pipeline, and then followed by we are making a metagenome metabolic model that that's derived from the annotation that was on this metagenome, and the tool that we use for the build metagenome metabolic model. And similarly, we'll make individual models for these bins. So once we have a draft metabolic models that was derived from genome annotations, and then we need to fill metabolic holes in these metabolic models. So this is the case even for organisms, model organisms like E. coli that I heard from other talks, like 30 to 40% of E. coli have hypothetical genes that we don't know the function of these genes. So there are knowledge gaps, and we need to fill these knowledge gaps with reactions. And so that's where the gap filling process comes in. And once we gap filled a metabolic model, then that model is ready for simulation. So we can do our predictions, and then we run flux balance analysis on these models. And then finally, we compare our metagenome model to its bins. And this is a very rich analysis that you can get out of the system right now, how we compare the entire metabolism of a metagenome and how it's distributed across all the bins. So that's the workflow today in a nutshell. And to to orient yourself and in the context of KBase apps. So what are the metabolic models, what they do? So the metabolic models metabolic models enable the rapid prediction of the metabolic capabilities of an organism, with the added advantage of being scalable with the amount of data that is scalable, that is available of the organism. So we have these organisms systems and we have these sequencing reads, and then we derive annotations from this sequencing reads. So the annotations means that we identify genes, we assign functions, and then based on those functions, we derive biochemical reactions. So very high level overview that you have heard the talks in this morning in very in-depth knowledge, in-depth detail about metabolic model, but very high level overview of what you see in a metabolic model for those who are less familiar with this concept. I would explain the metabolic model have primarily three components. That you have a metabolic network, of course, with that consists of compounds and reactions. And then we have a biomass this is what the cell is made of, that consists of cell wall materials, energy, cofactors, lipids, and nucleic acids, amino acids, things like that. And this typically the biomass is experimentally derived in metabolic models. And then the third component is, of course, a media formulation. So this is the light glucose minimal media. And typically in the biomass, it's known as the objective function because we measure the growth of a model. So you have heard talks in this morning about the concept of biomass. So the objective function of a model always doesn't have to be the biomass. It could be an excretion reaction that somebody could optimize the excretion of acetate or ethanol or another biofuel. But typically people want to measure growth. So that's why the biomass is considered a typical objective function. So why we use metabolic models? What can the metabolic models do? So the metabolic models is one of the great tools to understand the metabolism of an organism. And you can use the metabolic models to identify trophic interaction among organisms in a system, predict impact of genetic perturbations, identify media requirements in non-culturable bacteria. So there are bacteria that you cannot grow on experimental conditions in a laboratory, and metabolic models can help identify what's the minimal list of nutrients that you have to grow these organisms. And in the biofuel field, a certain organism can be used to produce an important biofuel. So also you can use metabolic models to predict culture conditions and possible responses to environmental changes. There are endless lists, and these are some of the highlights that I can put here to how metabolic models can be used. So we have to optimize predictions. So in order to optimize these predictions, phenotypic data are essential. So the phenotypic data means that more measurements need to be reconciled into metabolic models. And so like the metabolomics, so there's a very exciting talk right after this one, how we can use the cheminformatics where we expand the known biochemistry in generating novel compounds, and then map these metabolomic into the metabolomics data and identify these unknown peaks in metabolomics data and map to those compounds. Remember now, you have heard yesterday and day before, the FTICR data is a very high quality metabolomics data, but it has just the formula, not the structure. So what we are trying is to map these FTICR data into a potential new chemical structure if that particular compound is not known. So very exciting talk coming right after this and from Chris Henry. So look forward to that. And the more the measurements we have, the more the measurements that we can integrate, like from transcriptomic, proteomics, and from growth phenotypic data, the predictions will be more useful and more meaningful. So the metagenome model versus bin models concept. So as I explained earlier, so why these are important. So in a metagenome model now, we need to understand, so the metagenome model consists of the overall metabolism in the system. So we can answer scientific questions like what are the degradation pathways in this system? What anaerobic electron acceptors that this system able to or capable of reducing? Can the system capable of fixing carbon? Can we identify pathways related to metagenesis or methanotrophic behavior? So when you go to individual bin models, so these are like isolate organisms, genome scale models. So once we have these bins, then we can answer questions, we can address questions like what are the trophic interactions, what are the cross-feeding events. So in a typical system, one could fix carbon dioxide and excrete acetate, and another organism can use acetate as the carbon source. We can see these relationships when we have when we model these individual bin organisms, and the relative abundance of each member and their contribution in the system. So all those can be factored using the bin models. So this is also important that I got this numbers from Wrighton group, that there is also a reduction going from reads from assembly to the bins. So when you bin a metagenome, you lose some of the genetic information. So you may not see all the genetic information that's in the metagenome model. So that's also one important reason to look at a metagenome model and try to understand the overall metabolism. Because of the reduction, some of the pathways may disappear from these bin models. So in KBase, we have the built metagenome model tools that you can build a metagenome model that represent the overall metabolism. And we also have a compartmentalized community model, tools to build a compartmentalized community model. The tool is merge metabolic models into community model. This is where we take these bin models, high abundant bin models, the major players in a system, and then we can build a compartmentalized community model. And we can factor the relative abundance into this, and with a compartmentalized model, so each organism is a compartment, and where we can identify the trophic interactions, who's producing what, so who's the main carbon fixer, or who's have the degradation pathways for a given nutrient, and what is it excreting, and another organism use that excreted compounds as a carbon source. So those relationships can be determined from this kind of a tool. However, we are not using this tool in today's workflows, but we do use the metagenome metabolic model construction tool today in today's workflows. So so we have the metagenome assemblies and also the bin assemblies, and then we identify the functional annotations based on these assemblies using the RAST annotation pipeline. And then we build these metabolic models. And so it's important to talk about how we derive biochemical reactions in these models. So what we get from the annotation is that we have the annotation algorithms identify a feature like a gene, and then it's also assigns functional roles like pyruvate kinase. And then we have this complex, we have this concept of complex that for, in this case, the pyruvate kinase is a unienzyme that we map to a complex, and then we identify a biochemical reaction. So this biochemical reaction get added to the model. So in case of a enzyme that has multiple subunits like NADP transhydrogenase, so that comes from two genes, alpha and beta subunit, we map both these subunits to a complex, and we want to satisfy this complex, assume that both of these subunits are present. And if both of the subunits are present, then only we add this biochemical reaction into the model. And this is also helpful when we try to compute gene essentiality. So the gene essentiality computation is like one of the, when we simulate, when we do knockout simulations, if you knock out this particular feature here, that means that this complex will be no longer satisfied, and then we have to remove this reaction from the predictions, or avoid this reaction during the prediction. So the complex is really helpful to identify whether to see that all the subunits are present in order to add the biochemical reaction, and also when you do knockout simulations to remove a particular reaction from the models. And in the recent developments that we have add other ontologies into our model construction tools. Previously that we have to have the RAST annotations because we maintain a controlled vocabulary that we map these functional roles to the biochemical equations, but many people were requesting from the user community that they want to build based on other ontologies. So we have added that capability to our model construction tools, that you can build models based on KEGG KOs or EC numbers or metasec reactions in a prioritized order. So one could prioritize, okay, I want the RAST, look at the RAST annotations first, and then KEGG KOs or EC numbers. So one of the most essential components in our model construction pipeline is we want to get the ATP yield predictions right. So if you want to study the behavior of an organism, that's what the metabolic models are being used. And if you want to study the behavior, the ATP yield predictions, the energy prediction is very, very essential. And so we we put a special attention and effort to get the ATP yields predictions right. So we paid attention to the central metabolic pathways, all the fermentation pathways that we could find from the literature, and the electron transport chains that we can find from the literature. So as many of you know, bacteria is a mess. So they have a lot of anaerobic electron acceptors, and so we need to factor these into our metabolic model so we can predict, we can represent the energy metabolism correctly and also predict the ATP yields correctly. So right now we have all these electron acceptors able to capture the reductions of all these electron acceptors, and we are expanding that list to like sulfate, thiosulfate, metal oxidation, and so forth. So once we able to get the accurate ATP yield predictions, now then in our predictions you could see if the organism grows under aerobic conditions like E. coli, you can see the theoretical ATP yield is close to 26.5, where none of the electron acceptors are present, then the same organism then able to grow by fermentation, and at which point the ATP yield is close to 2. And then if you have anaerobic electron acceptors like nitrate or trimethylamine oxide, so then still you have the anaerobic respiratory chain is functional at that point, and you can generate slightly higher ATP yields. So these functions are right now available in KBase in beta mode app, and very soon will be released this app, so you can use as the released app. So we also worked on other improvements in our model construction pipeline. So we addressed the ATP yield, unrealistic energy biosynthesis pathways, and we want to improve the transporters of our model. And the transporter curation is a very complex process because the genome annotations are not clear and incomplete on many of the transporters. And also we want to capture the biochemistry from the public databases, which is also a very complex process. And because there's no controlled vocabulary that is, everybody developing these databases. So this is a TCA cycle map that shows that previously in our models that we weren't able to capture all the pathways and its fluxes, and this is just one example, but across the models now we were able to capture pathways, and we could accurately predict the flux distributions with these improved improved efforts into our model construction pipeline. So I'm not going to talk about the flux balance analysis. You guys heard in depth about flux balance analysis. However, in the context of KBase apps, so so if you have this kind of a toy model that these are the compounds and the letters are the reactions, that assuming steady state, so the type of the models that we are talking about today is a steady state models. So in a steady state model, no internal metabolites are allowed to accumulate nor degrade. So everything that converts to compound 2 needs to be converted to compound 3. So in that case, at steady states, the rates are equal. That you can see all the rates are equal here. And in this particular example, because most of the example that you don't see these linear pathways as my earlier demonstration, but most of the time you see these branched pathways, like glucose coming into the cell, that it can get oxidized in 3 different ways: glycolysis, internodeutrophil pathway, and another pathway that oxidize sugar. So in that way, it can distribute into 3 different pathways. So if certain nutrients enter the cell at the rate of 60, that you could see that this can distribute into multiple pathways, but the accumulation of these rates are equal. So the reason that I am explaining this is I want to come to these slides and explain about knowledge gaps and how this flux balance analysis fails if we have the knowledge gaps. So we could find knowledge gaps even with the well studied organisms like E. coli or B. subtilis. And so because we don't understand the whole metabolism, even for E. coli, 30 to 40% of genes, we don't know what they do, and it's worst in other organisms. So if you do have a gap, then the flux balance analysis or the predictions fails, and it doesn't work, as this compound cannot accumulate nor degrade. So we need to find these metabolic gaps in order to have a functional model. So that's what I'm showing here. So so I'm comparing here that how we gap fill, our approach of gap filling in a model in a rich medium versus minimal medium. So this slide explains how we gap fill organism in the rich medium. So in a rich medium, so these are the metabolic gaps that that's in the model, and in a rich medium, all these nutrients are present. And if that is the case, when the gap filling algorithm does it will add a transporter and import these nutrients into the cell and satisfy the biomass. So you would see lesser number of reactions get added to the model if you run gap filling on a rich medium, because it doesn't need to biosynthesize all these compounds in the biomass like amino acids, the vitamins, the fatty acids, because it can simply uptake these nutrients from the medium and satisfy the biomass. This is also exactly what's happening in the laboratory, and you could see that if you grow a cell in a rich medium like LB, it doesn't need to biosynthesize all the end components that it needs to satisfy the biomass, that it could simply uptake those nutrients from LB, and then the cell can grow. However, if you try to grow a cell in glucose minimal medium, then what happens? So every single biomass component, all vitamins, that has to be synthesized from that sole carbon source of glucose. In that case, it has to activate all its pathways and synthesize every single component that is needed to survive if it has the pathways. So similarly, if you gap fill in minimal medium, so then it will gap fill all these biosynthetic pathways towards the end products like the amino acids and vitamins and quinones and so forth. So if you gap fill in minimal medium, you could see more gap filling reactions that you will be able to see that today in your tutorials, and compared to rich medium. So where we find these gap filling reactions, so we fetch all the biochemistry that we can access from the public databases, and then we reconcile all these reactions into controlled vocabulary, and we then use these biochemical reactions to fill these gaps. So finally, so once we have the metagenome model, metagenome models, and also the bin models, then in today's workflows that we will compare your metagenome model across all the bins. So this is there's an app, the model comparison app that you could find in your tutorials. It's a very rich, useful analysis app that you could see this analysis. Like in this case, I'm showing that. So this is the metagenome model, and these are all the bins. And so I'm comparing now the gap filling reactions, and you could see, for an instance, that for the metagenome model has less number of gap filled reactions compared to the bins. The reason being that metagenome model is an accumulation of all these bins. So that's why the metagenome model has less number of gap-filled reactions compared to the bin, and some of the bins has lots of gap-filling reaction. That's also an indication that this may be a not good quality bin, that it needs to gap-fill ton of pathways in order to provide a flux distribution. So this app also gives some statistics, and so here it shows the number of gap filling that it needed in the rich media, and also the ATP yields. This is related to the central metabolism and ATP yields that I explained previously. And here you could see right away for some of the bins, the ATP yields are close to 26.5. So this indicates that these bins are aerobes, that they are capable of growing in the presence of oxygen and has a ATP yield close to 26.5, which is the theoretical yield of ATP yield of E. coli. And then you can see some organisms has the ATP yield close to 2, and this means that these organisms most likely to be anaerobes, strict anaerobes, that they can only fermentate and produce about 2 ATPs. And also there's another indication, as I mentioned previously, that if there's ton of gap filling, especially in rich media, that's also an indication that these are poor quality bins. So this can be also, these numbers also could be considered if you want to remove these poor quality bins for your downstream analysis. So with that, I would like to thank everyone who is involved, the KBase group, and everybody at EMSOL, and everybody at Argonne who helped to get this going. And now, if I have time, I would like to demo the narrative workflow so to get a better an idea. Becky, how much time I have?
It's now 10:43. You have about here until 11:15. So you have quite a bit of time.
Okay. So so this is the workflow today. So many of the students are very familiar with the KBase environment now, that all the narratives for today's narratives are added to the Microbial Dynamic Summer School organization, that all the narrative for today's narratives are labeled with the day 3, Summer School day 3. And you see here that it will give a description of what this workflow is about, and it will give you a menu that you can navigate to each of the steps. So you can click any of the steps that will go. And so what we are doing here is that we are importing a metagenome assembly into the narrative, and then we annotate that metagenome assembly with RAS-TK, RAS annotation pipeline. And this will give the RAS will identify the genes and assign functions, and you'll be able to search your favorite functions like pyruvate synthase, and this is a searchable table that should show up the annotations that you want to see. And it's really nice that the RAS also categorize these annotations into subsystems. So this is similar to the modules in DRAM that you have seen on Monday. So they categorize all the annotations into these subsystem and subsystem classes, which is very helpful to see the annotations in the context of these areas of metabolism. And then we built a metabolic model based on those RAS annotation. And so this is where I was explaining about the gap filling. So while we build the metagenome, we also gap fill these models. And so the gap filling reactions as a list is given here, and it will show this reaction either added, or sometimes existing reaction has to be reversed in order to get a flux distribution. So in this example, I see only the added is being mentioned here, but sometimes you can see the keyword reversed for some of the reactions. And this tutorials will give explanation of explanatory text for all the fields that you want to look for. So typically you will see all the reactions in the reaction tab. That this is also a searchable table. You can search the reactions, compounds, and the compartments. Typically that here we have the cytosol and extracellular compartments, list of biomass compounds, and the gap filling iterations that we used in this model, and also the pathways. So these are KEGG pathways that paints what genes are present and absent on the on different KEGG maps. So then we want to gap fill these models in minimal media. So in this case, we import minimal media formulations into the narrative, and then we gap fill in the minimal media compared to a complete media. So again, there's a list get added. So this is what I was mentioning earlier. Some of the reaction you could see that it was reversed instead of adding. So that was existing reaction in the model rather than adding, that it was reversed in order to get a flux profile. So then we analyze the individual bins. So in this case, we'll bulk annotate all the bins that related to a particular sample, and that's also we annotate with Rust annotation pipeline, and then we bulk build individual bin models in the system. And so in a typical FDA model, so again, the output table is very similar to what I shown earlier. You could see reactions, compounds, the individual genes that map to those reactions, and the biomass, and also the gap filling events. So then we run the flux balance analysis in the system, which creates a flux distribution. And here that you will be able to see the objective value, the growth, how much the growth is measured. And in this case, you can see the fluxes of individual equations. And also you see the exchange fluxes. This is where what compounds were excreted, or what compounds were uptake into the model. And exchange fluxes could be accessed from here. So finally, we compare the model, as I mentioned earlier. So we run the model comparison, and the model comparison is that we compare the metagenome model against its bins. And so this is a very rich app that it has many ways of that you can analyze your metagenome models against all the bins. And so check this out, and your TS will help to explain this app and how you can use to address your scientific questions. And then in the next talk, Chris will explain how we load the FTICR data into the system, and then expand the existing compounds into novel compounds and map these novel compounds to the FTICR data, which is I'm looking forward to be a very exciting talk. And that's all I have, and I'm happy to answer any questions.
Great, thanks, Janaka. So this is really exciting. I wanted to mention to all of the participants that the narratives that Janaka is showing here are going to be used by our students in our afternoon hands-on session, but they are available publicly on KBase as well. Again, you can find them by searching for Summer School Day 3. But we would encourage the public, if possible, to not copy the narratives and run them maybe until after the course ends, just to avoid overloading the KBase servers. But definitely those are available for you to look at and play with on your own as well. So have a number of questions that come in. So you're a little bit early. One of the questions, and I think you already addressed this, but why do you consider minimal media and not complete media at your first step of model construction? And I guess that's probably the first step of gap filling.
So the gap filling is a computational extensive process. So if we want to do iterative gap filling and find out what will be the minimal set of reactions to gap fill in a rich media, so that's why you could use the complete media first. So that the complete media is the rich medium that you can get. So in that case, what happens is that it will assess all the transporters in the model, and it will provide every nutrients that the model has the transporter for. So then if you want to gap fill in minimal media, you can do another iteration of gap fill in minimal media. And in that case, if you do iterative gap filling, it will add less number of gap filling and as an iterative gap filling. So then you can identify what's the difference between complete media and the minimal media, what additional reactions that was added when it grows on glucose minimal media.
Okay.
So it will give a further clarity between the rich media and the minimal media.
Okay. So another question that was, that people were talking about over in Discord is, if you could maybe explain a little bit about model curation. So as you're developing these genome models, where it's for a metagenome or for a bin or for an individual organism, and you're using these automated annotation pipelines, how much do you need to do any sort of manual curation of that, or what does that mean when you do curation of your model?
Okay, so that's a very good question. So when we so let me go to that. So when we annotate a genome and then derive a metabolic model, what you get is a draft metabolic model, and that has lots of gaps. So the systems give you a draft metabolic model, which is a good starting point for somebody to curate the metabolic model. So when we gap fill, the gap filling is based on, as I mentioned earlier, it's based on the media. So it's based on the media. So this is an algorithm, the optimization algorithm that's try to find the minimal set of reactions. So that's the gap filling strategy right now in KBase. We find a minimal set of reactions to fill these gaps. And these solutions, it could be wrong. So because there's lots of lump reactions in the databases, and also the strategies to identify the minimal set of reactions. So it's possible that there's a 8-step pathway that it can add like 4 lumped reaction to fill that gap. So the curation is very, very important, so that if you are the expert of a particular organism and you know the biochemical pathways well, the recommendation is that you would curate the model. So for an instance, so here we have an app called Edit Model. So using this kind of app, you can delete, add reactions that if you know the biochemical pathways very well. And it's very essential to get your metabolism, and if you know very well the metabolism of your organism, and to get that right.
Okay, yeah, that's great. Okay, so you can use the automated processes, but then you have some flexibility to add, say, expert information using the edit metabolic model as well.
Yeah, all the highly curated models, all the published models, they are all hand curated, and we have this tremendous effort right now. We are going through in our modeling pipeline and on the fungal model construction pipeline, actually, that we are working with PNNL uh, scientists at PNNL to improve this curation. So that's how all comes down to hand curation, time-consuming hand curation.
Right.
That there's no way avoiding that to get these high quality models.
And what role does direct experiments and measurements, like Mark was talking about in fluxomics, how does that play into curation?
So I refer to Chris if you are here to answer that question. So in generally, we could reconcile the growth phenotypic data or the expression data that we can reconcile. So based on the expression data, that we can turn on and off reactions based on the expression data. So if there's absolutely no expression for a certain gene, that the particular reaction we forced not to be active. And similarly for metabolomics. The fluxomic data also could be used the similar way, that if there's no data for in that particular environmental or media condition, that we do not activate those reactions. And so that's how the phenotypic data can be reconciled into models. Chris, if you want to add how the fluxomic data can improve the predictions, please go ahead.
I would say the thing to remember about FBA is that there's not one answer. The solution you get from FBA is a valid solution to optimize the given objective function, which is typically biomass. There are many different solutions that will give you optimal biomass production. So there's not one answer there. Fluxomics can help narrow that down, but here's the other thing. FBA optimizes biomass. Your organisms do not. Many of them come close, and that's why we use that as a proxy for, knowledge of what your genome actually does. Fluxomics actually, figures out based on your measurements what your organism is actually doing. So Fluxomics can actually figure out what the real flux is. FBA only approximates it based on some kind of objective function. And the thing to remember is that you might want to pick a different objective function to get a better answer for your genome, if you're in an environment where it's not optimizing biomass. So that's a critical piece of thinking when you're using this tool.
Okay, that's really interesting. So given that, Chris and Janaka, can you give us a little bit of insight, or maybe your own experience as to how well these kinds of models are actually able to predict observed behavior? So if you simulate a batch system and you control the environment, and you predict the amount of uptake of a certain substrate, how well in general in your experience, have these metabolic models performed?
Yeah, and for E. coli, this is what the seminal paper that announced that really described the use of optimization of biomass as an objective function for FBA was by Paulson. And what he shows in that paper is Bernard Paulson at UCSD. What he shows in that paper is that optimal biomass works for E. coli when it's growing on its own in a reactor with a lot of nutrient. It does definitely predict yields quite well for a lot of different organisms in a lot of different environments. People have used it to predict, biolog growth rates with some approximate, accuracy. It's been used to predict what Uwe Sauer will show you is that often organisms don't grow at the optimal rate, but they grow very close to it. So he calls it like, if you imagine the solution space as a piece of cake, organisms often grow where the objective function is towards the top of the cake. Organisms grow in the frosting. And that, I think it's a really nice visualization.
Yeah.
Yeah, it's a good approximation in many conditions, but in ecological conditions in soil is where, unfortunately, it starts to break down, because organisms, it's a very complicated, like, it's all about fitness, really, and growing fast doesn't necessarily maximize your fitness in soil.
Right. Yeah, that's definitely one of the complications we have to address as we start going towards ecosystem problems. Yeah. Here's another question that was in the Zoom chat, Janaka. So you talked about losing information when we go from a metagenome-based model to a bin-based model. So for the full metagenome, we tend to be able to use more of the genes, whereas or more of the reads, and when we assemble bins, we lose some of those reads. And the question was about asking if what we're really losing there is related to only rare organisms that occur less in abundance, or is there something else? And is there any way to avoid this in terms of bioinformatics?
So the one of the reasons that we losing this the genomic information is that during the bin process that they use some criteria like tetrahydronucleotide frequency or and other criteria to bin make these individual bins. And if a certain, whatever the criteria that this algorithm uses doesn't capture that genomic information, and that won't get into any of the bins, and it could be it's very low abundance organisms. And so, but I have seen that in some of the communities that even the low abundance organisms that they capture, some of they are low abundance, but the metabolism is fairly complete. And sometimes you see that pathways that occur in the metagenome and the reactions that occur in the metagenome model, but does not find that reaction in any of the bins.
Okay. Great. And a follow-up to the question just came through, including k-mers or unique k-mers to rare taxa can help avoid this problem?
I refer to Chris. Chris probably have a better idea of how the k-mers would able to avoid this.
What's the problem again?
The problem of losing information when you go to bin your metagenome.
And the question is like related to the k-mers, right? So would
Right.
k-mers contribute to avoid this problem?
There's a lot of things that contribute to this problem. The failure of assembly. And so you have a certain fraction of reads that get lost because they just don't get integrated in assemblies. That fraction is quite large in soil. It's maybe more like 40 or even 30% in gut. But there's that loss. You could potentially, and I've seen people propose this, but it's still sci-fi to me. Yeah, I mean, you can, of course, annotate raw reads with taxonomy and function, and then perhaps use that to recruit to bins. I think it might be possible in the future, but the problem is the best you'll be able to do is recruit to bins at a pretty low OTU specificity, and that's a problem. So you might be able to make like OTU level, like genre level bins that way, and sort of recover some of your lost reads. And the same sort of strategy could be applied to lost contigs, unbinned contigs. But it's always going to be approximate. The best you'll do is to, recover those things into genre or even family or order level, bins. And species level binning at the moment is really not possible. Maybe for some genomes that are extremely well surveyed in the reference space. But so I think there are some things you can do. There's no tooling that I'm aware of that supports any of that. You'd have to manually do it by mixing and matching a bunch of, tools that are out there, Kraken and MGRAST for functional annotation, or Kraken or Human2 for functional annotation, or Kraken for taxonomical annotation.
Yeah, I think if you look at the numbers Janaka is showing right now from the Wonders data from the different rivers, even just the straight metagenome assembly, at the most, it's getting 56% of the reads. So,
Exactly. It's I think the thing to remember is what is in that. I've looked at a lot of systems with this approach of assembly and then annotation and modeling. And what I found is that the species breakdown you get from the assemblies is not too far off of, like, here's a test you can run in KBase to kind of explore this. You could annotate with Kaiju or Kraken and look at your species distribution. And the nice thing about Kaiju or Kraken is they won't lose those reads. And then you could look at the coverage and species distribution you get from your bins and use GTDB to assign your bins to a taxonomical range. And compare those two and see, like, is your Kraken or Kaiju distribution wildly different from your bin distribution? And in my experience, admittedly, is mostly in gut. The answer is no. And so what you're getting out of the assembly is the highest abundance genomes, which is arguably a lot of the highest, abundance is going to translate to flux. There's going to be a lot of the highest fluxes in your environment. And so you could argue that's the most important thing. Of course, there are low flux processes that are very, very important, and you're going to have a hard time addressing those with assembly-based approaches.
right. Okay, great. Here's another question. So, and I'm not sure. Well, I'll just read it. Is KBase able to reconstruct metabolic network by orthology?
That question refers to using different ontologies?
I'm not really sure.
No.
Or propagation?
By orthology, I think what they're referring to is maybe our model propagation method.
Propagation, yeah, that's what I...
So you can import a model, any model you want in the KBase. The import tools are rough, but you can import a model, whatever model you want, built with whatever annotation you want, whatever chemistry you want. And there is a tool in KBase, if you just look up the word propagate, that lets you propagate that model to any other genome based off of orthology. And so you run the proteome comparison tool first to compute corresponding proteomes based on bidirectional best match. And then you run the propagation tool with that proteome comparison, and it will it'll translate the model you've imported to a new model. And that's actually how our current fungal model reconstruction tool works, essentially, is by that orthology kind of based translation.
Interesting. Okay.
So this tool can be used that anybody wants to build a model based on a published model that is out there, well-curated published model. And if your organism of interest is phylogenetically very close to that published model, the organisms that the published model was built, you can import that published model into the KBase and then generate a proteome comparison against the genome of that published model and of your genome of interest, and then you can propagate that biochemistry, building a new model for your genome of interest.
Now, I mentioned that our system doesn't do a good job with cyano and archaea, and so with cyano and archaea, I would definitely recommend using this app with a published with archaea model, of which there are many, or a published cyano model.
Yeah, so a follow-up question just came through on that. How close do the genomes need to be to use this propagation tool?
The closer the better.
my suggestion is try it out and see how it works. Look at your proteome comparison and think critically about that. So the proteome comparison tool tells you what fraction of the genes in your genome had a corresponding gene in the other genome you're comparing to. And if that fraction's pretty low, just be aware, like if only 50% of your genes match, well, at best, your propagation is only going to capture 50% of your genome. of the metabolic capabilities of your genome. Now, our propagation tool does let you use RAST and model seed to try to fill in, try to capture ad content from the other 50%, but still, that's just something to bear in mind. That's a good measure of how close you need to be.
So that's what this merge in KBase model, the functionality does. It will also do a traditional way of deriving the reactions based on the RAST annotations in addition to propagation.